7.5 million cases and 420,000 deaths. It seems that vaccines are the only way to end the COVID-19 pandemic. But why is finding a vaccine for the virus so challenging for scientists? The answer lies in the virus's genome.
To understand this, let us look at the concepts behind human body immune response. There are two aspects to every immune response: immunogenicity and antigenicity. When any virus tries to get through the cell membrane, the cell induces antibodies to protect itself. This is called immunogenicity. When these antibodies recognize the threat and destroy the virus, it is called antigenicity. Now back to the coronavirus. The SARS-CoV-2 genome encodes four major structural proteins that have the unique functions and properties. The spike protein is mainly responsible for the virus's entry to the host cells. The spike protein targets the ACE2 enzyme as the entry receptor, which is present on the surface of epithelial cells. The spike protein then uses its receptor binding domain to bind tightly with the enzyme. Then, a surface enzyme called TMPRSS2 activates the enzyme attached spike protein through a complex reaction. After this, the virus can enter the cell easily.
Now, ideally, when the spike protein binds with the ACE2 enzyme, antibodies should be induced to destroy the virus. Now, here's the catch.
To destroy any virus, the antibody should bind with the component on the virus called the epitope. In the coronavirus, these epitopes are found on the spike proteins. However, these epitopes are hidden away under a layer of glycans, due to which antibodies cannot bind to the virus. That means the antibodies think that the virus is just harmless goods. In short, antibodies are being released, but they are not fighting. This is the biggest challenge scientists are facing today, because vaccines only work when the immune system is able to recognize the virus. So what is the solution?
Even though the spike protein acts as a hindrance for treatments, it also acts as an opportunity.
Because the virus's entry is mainly orchestrated by the spike protein, it could be a potential treatment target. Such treatments could be in the form of drugs that forbids the binding of spike protein and ACE2 enzyme. Or it could be a drug that breaks down the layer of glycans to expose the epitope for the antibodies to bind to. Or even vaccines, which help the antibodies target the epitopes on the receptor binding domain, which is a minute portion not covered with glycans.
Now, while scientists all over the world are trying to find vaccines, there is still one factor that must be considered: time. The question's not how, but when. Because till then, all our lives are still at risk.